A virus is a non-living infectious particle. It consists of either DNA or RNA, which will either be single or double-stranded. There's a protein coat called a capsid that surrounds it. The subunit of a capsid is the capsomere. Some viruses will have glycoproteins, which will trick other cells into allowing it to enter. In some cases, viruses will only infect bacteria. These are called bacteriophages. Viruses reproduce in two different ways. One of these ways is the lytic cycle. The lytic cycle is where the virus is going to enter the host cell and take it over. It's going to turn that cell into a factory producing tons of the virus. Eventually, the cell will explode. This ends up killing the cell, but creating large amounts of the virus. Bacteria is able to defend itself by using restriction nucleases, which will cut up the DNA of the virus that has infiltrated its cell. Keep in mind, this reproduction cycle kills the cell. The lysogenic cycle is a longer process and does not kill the host immediately. What will happen is the virus's DNA will incorporate itself into the host's DNA genome. This will allow the host cell to automatically produce the proteins that will help the virus, and it will reproduce the virus when it goes through mitosis. The part of the virus's DNA that is implemented into the host cell is called the prophage. Both of these cycles are going to require a host, as viruses cannot reproduce on their own. The host range is what the virus can infect. All right, types of viruses. So there are animal viruses is the one that's commonly referred to. This has either DNA or RNA. RNA viruses, though, mutate more easily because there are no proofreading enzymes. If you guys remember, DNA polymerase III was checking its work after it replicated the DNA strand, and nucleases were present to cut out and fix any errors that were caught. In RNA, none of that is present, which is going to lead to tons more mutations in the RNA, not in the DNA. Retroviruses are made out of single-stranded RNA. They're basically going to do the reverse of transcription. They will enter the cell as RNA, but have reverse transcriptase enzyme, which will reverse the bases on the RNA and work itself into the host DNA. Once it is incorporated into the host's DNA, it is called a provirus. An example of this is HIV. Vaccines are focused on preventing viruses, so they usually consist of harmless forms of the pathogen, and they will be presented to the host. This will allow the host to know how to respond to the real virus by allowing it to easily kill or take out the harmless one. When it takes out the harmless one, it creates antibodies which can be used on the real one. In order to treat a virus, an antiviral drug is needed. This drug will interfere with the virus's replication. Plant viruses, these viruses affect plants. They're either passed down horizontally from one plant to another or vertically from parent to offspring. Viroids, these infect plants and interfere with their metabolism. Prions are proteins that are misfolded. While they aren't necessarily a virus, they have a domino effect and convert normal proteins into misfolded ones. Viruses and cancer. So viruses can change regulation factors put in place, which will cause cells to become cancerous. They will either turn the gas, like the quote-unquote gas on and unrestrained, or they will take out the brakes. An example of this is converting proto-oncogenes into oncogenes. Prokaryotes have 10 times the mass of eukaryotes. Bacteria can be harmful and helpful. A lot of helpful ones are in the stomach or ones in the environment that are involved with nitrogen fixation. Nitrogen fixation is a process of converting nitrogen gas into ammonia. We need this for protein and nucleic acids, as we cannot grab it from the atmosphere, so this allows us to utilize it. Some of these can be used in biological remediation to fix oil spills. Bacteria shape. It will either consist of a cocci shape, which is a sphere, bacilli rods, or spirilla, which is spiral. Prokaryotes have cell walls. They're made out of peptidoglycan. In plants, it is made out of cellulose. So while they do have cell walls, it differs from plants. Bacteria will have mechanisms allowing it to attach or stick. There are capsules, which are essentially like double-sided tape that will allow it to physically stick. The pili are structures on the outside that act almost like webs, allowing the prokaryotic cell to catch itself on other cells. Movement. Some prokaryotes have flagella, which is a long strand that physically moves, allowing the cell to move. Some have spirochetes, which use rotating filaments, which allow it to move. And then others have filamentous chains. These allow it to glide through swamp-like areas. Bacteria doesn't have a nucleus, so the DNA is in the nucleoid region. Antibiotics. So antibiotics can either be naturally or artificially produced. They inhibit the growth of bacteria by disrupting its metabolism. Nutrition. So prokaryotes will either be autotrophs, which means that they make their own food, either through light, which is photoautotroph, or chemoautotroph, which gets it through inorganic chemicals. Photoheterotrophs eat organic or carbon things that contain carbon. Chemoheterotrophs include saprobes, which decompose on dead organic matter, whereas parasites absorb nutrients from alive hosts. Reproduction. So binary fission is the process of reproduction in prokaryotes. Genes in bacteria can also be transferred. One method is transformation, where a prokaryotic cell picks a gene from its environment, pretty much just picks it up from its environment. Another is conjunction, is when a different prokaryotic cell gives a cell DNA. And then transduction, when a virus gives the cell DNA. Oxygen requirements. So some cells are obligate aerobes, meaning they need oxygen and cannot grow without it. Facultative anaerobes can switch between oxygen and no oxygen, but they grow better with oxygen. And then obligate anaerobes, oxygen is poisonous to them. Evolution. At first, cells didn't use oxygen. Once oxygen accumulated, eukaryotic cells utilized this. The first prokaryotic cells were heterotrophs. And it is believed that the first genetic material was RNA, but DNA became preferred as it had less errors in it due to its proofreading processes. Early Earth conditions were very extreme. That's why when the first prokaryotic cells emerged, they were archaea. As oxygen became to accumulate, there became more cells that are going to utilize oxygen. Prokaryotes can be split into two groups. There's archaea and bacteria. Archaea were the earliest cells. These can live in very extreme temperatures. Some of them include methanogens, which produce methane as waste. Extreme thermophils, which like hot environments, and then extreme halophils, which live in extremely salty environments. Biotechnology. So biotechnology has many applications in today's world. Many scientists genetically engineer or manipulate the DNA of organisms. Cloning. This allows scientists to produce large numbers of a gene or protein. Scientists can use this to examine the DNA. Lots of tools will be needed in order for scientists to do this. Vectors. So vectors are a DNA molecule. They are often plasmid or virus that is used as a vehicle to carry a particular DNA segment into a host cell as part of a cloning or recombinant DNA technique. Restriction enzymes. So these are enzymes that will cut into DNA fragments. They will cut DNA at recognition sites, which will be identified by a palindromic sequence. A palindromic sequence is a sequence of letters that can be read forward and backward and display the same thing. The restriction enzymes won't cut directly through like scissors. They won't always cut directly through like scissors. As you can see here, the process can result in either sticky ends, where it leaves part of the strand cut with unpaired bases, or blunt ends, where it cuts directly through, or essentially a clean cut. If a restriction enzyme cuts in the middle of a gene, the gene won't be able to function. Additionally, if the gene was cut out, and inserted in the middle of a gene sequence, the gene that it was inserted into will be useless and not function. Ligase enzymes, these are enzymes that fuse the DNA fragments together using ATP. Plasmid. So a plasmid is a genetic structure in a cell that can replicate independently of the chromosomes. Essentially, it is not part of the bacteria's chromosomes, but can be taken in by the bacteria. Scientists often will use this to transform bacteria. And yeah, so transformation is a process of bacteria accepting a plasmid. All right, so the steps of making a recombinant plasmid. First of all, the plasmid has to be isolated. Restriction enzymes will have to cut out the piece of DNA that you want to insert and cut out the area of the new plasmid in order for that DNA to fit. The DNA has to be inserted into the plasmid. After it has been inserted into the plasmid, ligase will fuse it back to its continuous state. Keep in mind, if you are inserting DNA from a eukaryotic cell into a prokaryotic cell, introns must be removed, as prokaryotes don't have spliceosomes to remove them. Once you have made the recombinant plasmid, you must introduce it to the cell you want to transform. To allow the plasmid to be inserted, scientists will often heat shock the membrane. Additionally, scientists can use electroporation or electrical pulses to create holes in the membrane to allow the plasmid to enter. Then you can observe the cells growing and eventually identify which ones have been transformed. During cloning, scientists can encounter some problems, one of which is expression. To counter this, expression vectors, which carry a promoter to express the gene, are inserted. Another problem is editing. In order to fix this, cDNA is used to clone eukaryotic genes into prokaryotes. It is a version of the gene with no introns, as prokaryotes can't get rid of them. It is created using reverse transcriptase. It's used to produce proteins in a cell that are not naturally developed by the cell on its own. In eukaryotes, the cloning process has to begin by grabbing a fertilized egg, which is the first stage in the embryo process, and removing the nucleus of that fertilized egg. By removing the nucleus, you can get rid of all the existing DNA. To clone a eukaryote, you must grab a somatic cell of the eukaryote you want to clone, take the nucleus out, and insert it into the fertilized egg that I just referred to with no nucleus. This will allow the cell to multiply in mitosis, but with the existing DNA of the organism you want to clone. PCR. This is a copying process of DNA quickly without using live cells. It can start with the insertion of a small amount of DNA. The machine will heat up, causing the double-stranded DNA you inserted to split into two strands. After this, primers in the environment will begin the DNA replication process, with DNA polymerase III following behind it. Essentially, PCRs are going to artificially replicate DNA multiple times. It can be used to amplify samples of DNA and to allow scientists to run tests risk-free without damaging the little amount of sample they have. Electrophoresis. Gel electrophoresis separates DNA, RNA, or proteins based on size by cutting it up with restriction enzymes. It is inserted into a container and charged using an electric field applied to a gel. The results show bands that represent the different fragments or molecules, allowing analysis of their size and quantities. The purpose of gel electrophoresis is to be able to compare DNA. If the results are very similar in terms of where the DNA, RNA, and proteins lined up, there's a high chance that the DNA came from the same organism. This can be used in crimes and maternal testing. DNA sequencing. There are two methods. So one of them's called the slow method, which involves sequencing DNA fragments individually, whereas next generation sequencing enables simultaneous processing of multiple DNA fragments, resulting in substantially faster sequencing. Genomics, so proteomics studies proteins living in living organisms. Bioinformatics, it deals with biological data analysis, and SNPs are tiny genetic variations. Stem cells. So stem cells are versatile cells that can turn into different types of cells in the body. Totipotent cells can become any cell. Pluripotent can become many, and adult stem cells are more limited but still help with tissue repair. They're important for potential treatments and understanding diseases.